in bioinformatics analysis tools used in SEAPharm research, 100 Pharmacogenes PKSeq panel. And it is our greatest honor and pleasure to have another honorable speaker that will be sharing on this topic of discussion. That I would like to firstly inform all ladies and gentlemen that all of you are highly encouraged to share your comments or even your questions so that we can together elaborate more on the topic of discussion for the benefits of the other audience, ladies and gentlemen. That is the reason why I'd like to take this very opportunity to introduce to all of you the representative from Riken Center for Integrative Medical Sciences, Dr. Koya Fukunaga.
One.
Yeah, so I'm sorry to delay my presentation. So now maybe you can see my presentation. So time is so small. So please let it begin my presentation. So today I would like to talk about an example of pharmacogenomics and the Riken targeted NGS panel. I named this target sequencing PKSeq for stratified drug therapy. In the first half, I will show the details of bioinformatics analysis of PKSeq. In the latter half, I will show the result of the SEAPharm project after sequencing the genomes using a PKSeq. First of all, I would like to introduce pharmacogenomics, but many people know the pharmacogenomics, so you are a pharmacogenomics specialist, so I will talk about shortly. So when a medicine is administered, a small proportion of individuals may experience adverse drug reaction, such as Stevens-Johnson syndrome, and leukopenia, and alopecia, and so on. Therefore, there are two groups of patients, those who experience ADLs and those who do not experience ADLs. We aim to determine which group a patient will be in before they receive a medicine by identifying the genetic variations associated with ADLs. This is PGX. So therefore, PGX is the study of how the genetic variants affect a person's response to drugs. Next, the Pharmacogenomics Research Network, PGRN, was re-established in 2015. PGRN carefully considers the guidelines for the clinical implementation of PGX. This is a list of PZX guidelines issued by PZRN. These are some of the most well-known biomarkers. Importantly, almost all biomarkers were derived from many pharmacokinetics genes, like this 75%. Therefore, we targeted the pharmacokinetic related gene, not pharmacogene. Pharmacogene contain pharmacokinetic and pharmacodynamics. So in this study, we targeted the pharmacokinetic related genes to discover the biomarkers for drug response efficiently. After that, we developed the sequencing method for all coding regions of the genes. NGS instruments have significantly improvement the output of automated Sanger sequencing instrument used in the Human Genome Project. It is important to note that some instruments may outperform Sanger sequencing with respect to read length. Currently, NovaSeq can produce sequencing data consisting of the 30 genomes in 2 days per one flow cell. Therefore, we can conveniently perform WES and WGS. General sequencing process utilized in NGS platform. I will show the simple workflow of a targeted sequencing panel for 100 pharmacokinetic related genes. I categorized the PK related genes depending on the function, drug metabolizing and drug transporter, and others. Using a multiplex PCR in combination with NGS, we can detect common and rare variants in all coding regions of the PK-related genes. This lists the detail of 100 pharmacokinetic-related genes, including 60 drug metabolizing enzymes, 37 drug transporters, NUDT1, 15, and VKRC1. We would like to show the workflow of a PKSeq in details. At one sequencing run, we can sequence 96 samples. We performed the multiplex PCRs of 8 times without using a commercially available library kit. Therefore, we don't need the cost of a commercially available library kit. So a little lower cost. To distinguish the 96 samples, we attached the individual adapter to PCR product. After the measurement of library concentrations, we sequenced this library using Illumina MySeq instrument. We selected the MySeq reagent kit of 500 cycles. So read length is a 2 by 250 base pair. After we obtain the FASTQ files, we analyze them using the workflow based on the GATK best practice. Briefly, at first I trim the low QC base using a Trimomatic software, and I use BWA and GATK. After that, we obtained the SNV, indel, and CNVs. We would like to show the to call the variants. I used the general bioinformatics analysis such as GATK best practice. However, please be careful of lead trimming steps. To remove the adapters and And to remove low quality bases is a general flaw. However, do not remove the primer sequences in trimming step, because primer sequences include gene-specific bases. For example, look at the down panel. There are the sequences of CYP2A families. To amplify the only CYP2A6 sequences, I used the gene-specific primers like this, like a forward primer, except of this base. All bases between CYP2A families are the same. If you remove the primer sequence, we could not map the leads correctly. Therefore, we must not remove the primer sequence from raw lead to map the reference genome. From this slide, I would like to show the higher accuracy of PKSeq than those of whole exome sequencing and whole genome sequencing. WES and WGS cannot map a part of the regions in 100 PK-related genes due to high homology sequence. This slide showed an example of mapping errors depending on lead lengths of the CES1 genes, carboxyl esterase 1 gene. General lead lengths of WES and WGS are 2 by 100 and 2 by 150 base pairs, respectively. As was apparent, the result after mapping lead to reference genome by each platform showed that WES and WGS cannot distinguish CES1 and choosing over CES1. Actually, in these regions, no variants are correct. You can see PKSeq data. In other words, these results suggest that short read cannot be mapped to reference genome correctly. Next, I will show another example of mapping error in WGS. I'm showing CYP2D6 mapping data of 3 individuals in WGS and PKSeq data. You can see the variants from WGS data in CYP2D6 exon 4 of WGS data. These are error variants. When CYP2D7 exon 4 had variants, the read map to CYP2D6 exon 4 like this of WGS data. When we calculated the total genotyping count of this synonymous substitution from this WGS data, no variant homozygosity were detected in 104 Japanese individuals. From the genotyping count, you can understand that these variations were mistakes. This error is related with CYP2D6 star 4 core variants like this. If the primer did not amplify the CYP2D6 specific region, we could not determine the heterozygosity variations. However, using the same primer set and I sequence SEAPharm UAE data, you can see that the heterozygosity of CYP2D6 star 4 and amino acid substitution could be detected. Therefore, we showed to be able to develop the gene-specific primer sets. On the other hand, after we sequenced and one individual genome using WES and WGS, we compare these results with those of Sanger sequencing data. Following this, we calculated the accuracy of WES and WGS with Sanger sequencing using the same individual. REF refers to reference allele, and out refers to variant allele. As a result, several discrepancies were observed in comparison between WES and Sanger sequencing, leading to lower sensitivity. Therefore, we needed a high accuracy sequencing method other than WES and WGS. Using this sequencing, we sequenced the same one individual genome. After that, we compare accuracies of a PKSeq panel with Sanger sequencing using the same individual. As mentioned above, REF refers to reference allele, and the out refers to variant allele. As a result, PKSeq showed completely similar genotypes as that noted with Sanger sequencing. Using this sequencing and the Japanese grant, we sequenced the 990 Japanese individuals. Subsequently, we registered all genotypes of these variants in the NBDC, Japanese Human Database, for the future PZX. Now, every researcher can see these data. Next, I'm showing the novel variation from this Japanese data. Yellow panel shows the novel amino acid substitution. Although many researchers have discovered PK-related variations for many long time, we can still detect the novel variants. Therefore, PKSeq panel will be useful to discover the novel variants associated with drug response. I show the PharmBar website. PharmBar means the Pharmacogene Variation Consortium, which is a central repository for PZX variation that focuses on haplotype structure and allelic variants. The information in this resource facility basic and clinical research, as well as the interpretation of a pharmacogenetic test, result to guide precision medicine. Like this, I registered the novel haplotype of CYP2D6, like this. As I introduced them in the previous slide, I sequenced 990 individuals using a PKSeq panel. The history to discover the CYP2D6 variants is very long, and many researchers continue to sequence many samples to discover the CYP2D6 variants. Nevertheless, we found the 14 novel variations in 990 individuals. This is very amazing. For clinical implementation of these variations, we must determine the haplotype between these novel variations and the non-functional variations as the next step. Here, a problem has occurred. To detect the haplotype of CYP2D6, read length of a short-read sequencer is not enough to determine the haplotypes between variations. Therefore, I used the long-read sequencer to detect the haplotypes of CYP2D6. This time, I used the sequel of a PacBio company. Although I did not show the data in this slide, I detected the novel 10 CYP2D6 haplotypes. However, CYP2D6 star 36 carried on two novel CYP2D6 variants. CYP2D6 star 36 is a no-functional allele. Therefore, these variations are no effects on CYP2D6 enzyme activity. When we detected a novel variation, we sometimes judge to carry the novel variation on CYP2D6 star 1 wild type without detecting the haplotype. As shown in this slide, novel variations did not always carry on wild type such as pathogenic variants. Therefore, when we detect the novel variations, we must confirm the haplotype between novel and known variations. Next, I will introduce CYP2D6 genetic diversity in Japanese, which is one of the most implemented biomarkers for PZX. I sequenced 189 genomes in Ryukyu populations. Ryukyu is Japanese subpopulations using a PKSeq panel. And I compared the frequencies of CYP2D6 genotypes and non-phenotypes in the Ryukyu and HOND subject. HOND means except of Ryukyu. X-axis shows the genotypes, and the y-axis shows the frequency. White bar shows the HOND frequency, and the gray bar shows the Ryukyu frequency. And the UM means a high CYP2D6 activity, and the EM means a normal, and the IM means a half activity, and the PM means a low CYP2D6 activity. Finally, I summarize the frequencies of CYP2D6 phenotypes in pie chart. Interestingly, UM and EM frequencies in Ryukyu subjects showed higher than those of HOND cluster. Therefore, drug response in Ryukyu subject might be different from a HOND cluster. As shown in And next, from this slide, we will show the application example using PKSeq. As you know, there are Southeast Asian Pharmacogenomics Research Network. In SEAPharm project, I'm involved in SEAPharm resequencing project using a PKSeq panel. This slide shows a number of samples in a SEAPharm resequencing project using a PKSeq. To predict the risk of drug response and adverse drug reaction using a pharmacogenomics biomarker, measuring general allele frequencies is very important. However, we do not know the earthquake. Earthquake in Japan. So, sorry. No. Now, however, we do not know the normal allele frequencies among these countries because of low accuracy of the NGS platforms. Therefore, Dr. Wasan's proposal is promising for future precision medicine. I already showed the accuracy of PKSeq. So almost all variants of PKSeq were concordance with those of WGS. Therefore, to confirm the accuracy of SEAPharm variants detected by PKSeq, I compared the allele frequencies between PKSeq and WGS using the data of SEAPharm project and the genome aggregation database. This is a very big database in Europe, abbreviated as GenomeAD. To compare the frequency, I used the allele frequency of East Asian population in genome AD. R square of allele frequency between PKSeq and the genome AD showed high values like this. Therefore, SEAPharm data suggested a high accuracy. Next, I will show the different variant types in 9 populations of a SEAPharm project, including 5 sub-populations, 5 sub-Indonesian populations. In PK-related genes, the proportion of non-synonymous variants were higher than those of non-synonymous variants. I want to say the non-synonymous variant is high. Next, we annotated the high quality variants using a public database, including DB-SNP version 150 and genome AD. Most of the coding regions were singleton or very rare, doubleton and tripleton. Dark blue panel shows the proportion of known variation, and light blue panel shows the proportion of novel variations. However, the absolute numbers of novel variations with a minor allele frequency less than 1% within the SEAPharm database is large. Of variations with a minor allele frequency more than 5%, we did not see the novel variants in DB-SNP. I compared the genome AD, so the tendency is the same. I'm showing the novel variations not registered in PharmBar from Japanese and SEAPharm data. Yellow panel shows the non-amino acid variations. Red panel shows the novel amino acid variations. As shown in Japanese database, many novel variations were detected. SEAPharm data project is the same tendency. So this data is a very precious process data. Oh, it's time. Next, I detected or estimated the haplotype of all PK-related genes. For example, I show the NAT2 alleles. NAT2 gene is very small. Exonic region is less than 1,000 base pairs. Therefore, we can directly detect the haplotype using MySeq reads. At PKSeq, we used the 2 by 250 base pair reads. I combined both reads and show the mapping status using IGB software in this slide. In the upper panel, this subject carried on NAT2 star 5 and star 6. You can see blue base and red base carried on the same chromosome like this. And the red base and the green base carried on the different chromosome. Therefore, this NAT2 genotype is star 5 and star 6. Using this strategy, I showed the global distribution of NAT2 phenotype from NAT2 genotype in SEAPharm data. As we can see, Greece, UAE, and India slow accelerator showed relatively higher frequency than those of the Southeast Asian countries. Surprisingly, Laos and the Indonesian slow accelerator showed a higher frequency than the other Southeast Asian countries. Japan and China slow accelerator showed very low frequencies. Therefore, these data suggested that the slow accelerator frequencies of SEAPharm were middle type compared with European and East Asian populations. These are the very precious findings. So this is the summary. So it's time. So this slide lists my collaborators. I thank all of collaborators in SEAPharm project. Thank you for your attention.